Xenia Mohanta, and I'm a senior postdoctoral fellow at Johns Hopkins School of Medicine. And today, the topic that I'm talking is nanomaterials for real-time biomedical applications. So, um, before going to my presentation, I would like to talk about my journey. I did my BTech in Applied Electronics and Instrumentation and MTech in Applied Electronics and Instrumentation with specialization in VLSI and embedded systems, where my thesis was on design and characterization of an optical glucometer. And then I went ahead and did my PhD in biomedical engineering in 2014 from Indian Institute of Science. And my thesis was on engineering graphene oxide surface chemistry and investigating its effect on the physicochemical and biological properties. And currently, I am doing my postdoctoral fellowship at Johns Hopkins School of Medicine, Kennedy Krieger Institute, and in the Department of Radiology. The topic that I work on is CEST MRI pH mapping using small molecules and murine models for chronic kidney diseases. Apart from this, I'm also working on another project parallelly here, which is on gene monitoring using reporter genes. During this entire journey, this is just my educational journey that I have shown here. But apart from this, during the year 2010 to 2012, I have also served as an electronics and physics lecturer at Royal College of Science and Technology in Bhubaneswar. And I have also had short stints as assistant professor in SOA University as well as C.V. Raman College of Engineering in Bhubaneswar. So for all the fellow faculties, I understand all the struggles and all the necessities that you require to go through this entire journey. Been there, lived there, and yes, and after that, after my assistant professorship stint, I have went on to do my PhD. I'm sure a lot of PhDs must also be here. I have kept the entire content very generic. So if you want to learn about something in detail, I'm happy to answer your questions. You can connect to me on LinkedIn. And the content of my talk today will be introduction to nanomaterials, then types of nanomaterials, real-time biomedical applications. I'll be focusing only on 2 case studies and impact, and then talk about the challenges and future directions, which will be the most important thing for you, of course. Starting with introduction to nanomaterials, I heard in the previous session that a lot of faculty here are not from engineering background. A lot of people may not know about nanotechnology. So I would start off with very basics, but I'm going to really breeze through it very quickly. So when we say nanoscale, we mean the nanomaterials. We mean the materials that have at least one dimension smaller than 100 nanometers. It has unique properties such as the increased surface area, and then it has quantum effects because of which the optical, electrical, and magnetic properties are altered. There is enhanced reactivity, so their chemical reactions are higher. As I said, it has unique physical characteristics. Now, optical properties, it has the surface plasmon resonance in metal nanoparticles especially, which enable applications in imaging and detection. It has great mechanical strength. In fact, graphene layer of graphene is considered as one of the toughest materials known to man, often exceeds that of conventional materials, making them ideal for engineering and biomedical applications. For chemical and biological properties as well, when you go at nanoscale, the chemical reactivity is very high, and that is because of the high surface to volume ratio, and thus it increases reactivity. The biological interaction also increases because of the higher and increased surface area, and thus
ಓಪನ್ ಆಗಿದೆ ಬಟ್ ನಿಮ್ ಹೆಸರದು ಏನು ಗ್ಯಾಲಾಕ್ಸಿ
Ma'am, you are muted.
Ma'am, you are muted.
I've been talking endlessly. Have you been hearing me?
Yes, ma'am. Yes, ma'am. Just
Okay,
for the
I
last 10
think
seconds.
yeah, I think I got muted along with everybody else, but that's fine. Okay. So nanomaterials has been revolutionizing medicine because of its versatility in applications. It has found its application in cancer treatment, in cardiovascular therapies, regenerative medicine, and thus has become integral to healthcare innovation, treatment, and solutions. It has revolutionized diagnostics and therapeutics. In diagnostics, it has been used now to find earlier and more accurate disease detection, enhanced imaging technologies such as MRI, PET. In fact, there is an upcoming imaging technique. I'm sure a lot of you must have heard about MPI. And then there's biosensors for real-time monitoring of biomarkers. In therapeutics, it has been found that nanomaterials provide precise and effective drug delivery. There is reduced side effects and improved patient outcomes. Stimuli responsive systems for controlled drug release have also been developed. So in relevance to biomedical applications, it plays a very pivotal role in medical research. It has found its place in nanomedicine, in personalized healthcare, and it is adaptable to existing medical technologies, advancing healthcare solutions. So I'm particularly talking about real-time monitoring applications here, and thus one of the examples that later on that I'll be talking about is continuous health tracking through nanosensors. So there are wearable devices which have been developed which can incorporate nanotechnology for diagnostics, and then it can be integrated with AI for real-time data analysis and decision making. So overall, I would like to say that nanomaterials is a very booming field, and in the past decades, we have seen various applications of nanomaterials. Although there are some challenges which lie in front of nanomaterials, but we see several developments for nanomaterials, and it has revolutionized the field of medicine. What are the different types of nanomaterials? Again, it may be very basic, but based on structure, based on composition, based on origin, and based on functional properties, nanomaterials have been classified into different categories. Based on structure, it is zero-dimensional, one-dimensional, two-dimensional, and three-dimensional. The dimensional structures depend on how many dimensions of the nanomaterial are in the nanofield. For an example, zero-dimensional are quantum dots. For an example, one-dimensional are nanowires, nanorods, nanotubes. Two-dimensional, there are nanosheets like graphene and graphene oxide. And three-dimensional, they are nanostructured bulk materials with interconnected nanoscale features. For an example, in polymers. Based on composition, there are metal-based nanomaterials like gold, silver, and iron oxide nanoparticles. In fact, if you search about nanomaterials and when you think about nanomaterials, I believe that gold, silver, and iron oxide nanoparticles are the first nanoparticles that come to your mind. Carbon-based nanomaterials such as graphene, carbon nanotubes, and fullerenes, polymeric nanotubes, similar as three-dimensional nanomaterials that I was talking about, they are biodegradable, and synthetic polymers like PLG and PEG, ceramic nanomaterials like silica, zirconia, alumina nanoparticles, and hybrid nanoparticles, which can be a combination of the organic and inorganic components. For an example, core shell structures. One of these core shell structures is, let's say, iron oxide with zinc oxide as a shell. So there are various topologies which have been designed, various examples of these nanomaterials based on compositions. Based on origin, there can be natural nanomaterials, there can be engineered nanomaterials, there can be incidental nanomaterials. Incidental nanomaterials are byproducts of industrial processes. And based on functional properties, it can be magnetic nanomaterials. And this is what I have been working on. So this is a very dear nanomaterial for me because I have been working on MRI contrast agents since my PhD. Worked on graphene oxide as MRI contrast agent. And right now, although I'm working on small molecule based contrast agents, I still stay associated with MRI contrast agents. So this is a very dear field to me based on the functional properties. Optical nanomaterials, for an example, quantum dots for fluorescence imaging, and then catalytic nanomaterials which act as catalysts in chemical reactions. So going ahead with real-time biomedical applications, that's what we are talking about. That all the basics that we heard about nanomaterials now, but how do they find their applications in biomedical application? Because you must have heard that nanomaterials can be used for optical sensors, they can be used in photovoltaic cells. So let us see how these nanomaterials can be used in real-time biomedical applications. So one of these examples, larger categories, I must say, is diagnostics and imaging techniques. Am I going too fast? Please tell me. If I'm going too fast, I'll try to be a little slow. I mean, anybody could answer. This is okay?
Ma'am, it's okay.
Okay, madam.
No,
It's
madam.
okay.
Yes, ma'am. Yes, ma'am.
It's okay. Space is okay.
Please go ahead.
Okay, thank you. So on a broader field, when we talk about diagnostics and imaging techniques, it is one of those fields which is very, very, very integral of the medicine field. Because whenever we go and talk about, let's say for an example, as cancer treatment, then you think about the first step is diagnosis. And you always think that how early diagnosis can be done so that, you can know whether there is cancer or not. It's as simple. It may sound simple, but when you are developing these systems, there are so many challenges while you are developing reproducibility, repeatability, trying to get the thing right, trying to get the right size. There are so many challenges that you face. So let us start with this biomedical application, which is diagnostics and imaging techniques. So one of these examples is quantum dots in real-time imaging for fluorescence imaging. So quantum dots are semiconductor nanocrystals that emit specific colors based on their size. Their stability and brightness make them highly effective for real-time imaging applications, facilitating tracking of cellular processes and disease progression in live organisms. As you see in this figure, the color that you get for different quantum size changes with the size. So as you go from very small to larger, it goes from the lower spectrum, that is blue, towards red, higher wavelength. So this is just one example of diagnostics where quantum dots can be used. But quantum dots still now have mostly been used in diagnostics, and you would see when you try to find out the applications, you can go to Thermo Fisher or some other website where they are actually selling these quantum dots probes, Q dot probes as they call them. You'll see that they can be used for Western blots, they can be used for fluorescence imaging, they can be used for various imaging techniques where you are trying to diagnose or find out that what kind of cell is there. You tag quantum dots along with markers, biomarkers. So quantum dots alone, they are useful for fluorescence imaging, but when you are trying to find out any biomarkers, active bioreceptors, then you need to tag quantum dots with active biomarkers according to your application.
Excuse me, but what type of element is used for this quantum dots, ma'am?
So you start with graphene. Quantum dots are mainly made from graphene. So
okay.
Yes.
Okay, thank you, ma'am. Go on.
Yeah, so you need to do these chemical reactions in order to get these quantum dots. In fact, it's very interesting, right? For quantum dots, they even got the Nobel Prize in chemistry, isn't it? So it is one of the greatest discoveries in terms of nanomaterials, if you look at it. And in real life application, quantum dots you can find even in QLEDs, right? Quantum LEDs. So when you look at semiconductor industry, quantum dots have been used in electronics a lot, but it also has a lot of application in imaging as well. So going ahead, iron oxide nanoparticles for biosensing. I have personally developed these iron oxide nanoparticles during my PhD, so I can tell you in and out about these iron oxide nanoparticles, that how versatile they are. Depending on the type of chemical reagents that you use, you can vary their size, you can do different kind of reactions, you can make them biocompatible with PEG. In fact, if you know about magnetic particle imaging, MPI, which is one of the latest imaging technique, so in that it is completely based on iron oxide nanoparticles, and these are it's not yet FDA approved. FDA approved iron oxide nanoparticles are used for MRI, but MPI hasn't been used in humans right now. It is still in preclinical studies. So you would like to learn about MPI, which is a really developing imaging technique right now. And so iron oxide nanoparticles is used for MPI, in MRI as well as T2 contrast agent, and they can, they can serve as contrast agents in MRI and are utilized in the detection of biomarkers, enabling rapid and sensitive diagnostic results. So what researchers have done over the time is they have modified the surface of iron oxide nanoparticles, conjugated it with different biomarkers, be it DNA, mRNA, and then they have used it for tagging and trying to attach it to different bioprobes inside the body. So they are different. You will find a lot of papers on iron oxide nanoparticles conjugates. So this is what a nanoparticle, iron oxide nanoparticle would look like when you look through a TEM, transmission electron microscope. Going ahead, another broad category of real-time application is drug delivery and therapeutics. And one of those examples is targeted therapies with nanoparticles. So when I talk about targeted therapy, when I talked about quantum dots as well as iron oxide nanoparticles, in fact, I just give two examples. There are just two examples. There are many more examples. There can be gold nanoparticles as examples. There can be gadolinium oxide nanoparticles as examples. Now, all these different nanoparticles, how do you actually get them to do these drug deliveries and therapeutics? So when I'm talking about targeted therapies with nanoparticles, you are going to actually modify that surface in such a way that you can attach these different moieties on top of it. It can be transferrin, it can be for transferrin receptor, it can be for folic acid receptors. So for folic acid receptors, it's going to be folic acid that you're going to attach on top of it. Sorry. So how is it useful? Cancer cells, I'm taking cancer cells as an example because in many papers where you talk about drug delivery and therapeutics, they are mostly cancer oriented. So I'm talking about cancer treatment, what they do is these transferrin, VCAM, folic acid receptors, lectin receptors, in fact, alpha beta integrins, VEGFR, these are all overexpressed in cancer cells. Since they are overexpressed, it's not as if these are not present in normal cells. These are present in normal cells as well. But I'm using the term overexpression. So when you are saying overexpression, as a result of which there is more expression of these receptors in cancer cells, as a result of which, let's say if you have a nanomaterial on which you are coating with, for an example, I'm just saying folic acid for an example, and you're coating it with folic acid, and there is cancer cell which has more folic acid receptors overexpressed. So as a result of which these folic acids attach more to the cancer cell, and thus you are able to detect these particles. It can be through imaging moiety modality, for an example MRI, or it can be fluorescence imaging, and then and you can also obtain targeted therapies. For an example, in PDT, a photodynamic therapy or photoacoustic therapy, in case of photothermal therapy, PTT. So in all these cases, you are trying to get the drug to the cancer cells, for an example. Now, in that case, what you are doing is, there are three components now.
Excuse me.
Yes.
Ma'am, excuse me. Ma'am, what is the percentage of success of this diagnosis, that is cancer diagnosis, in the earlier stages, ma'am?
Okay, so in the earlier stages, first of all, in the earliest stages, one of the greatest challenges that you face is early detection. How many people have you really heard that in stage 1 or stage 2, the cancer has been detected? It is very rare.
Yes,
it is extremely rare. So these, when I'm talking about biomedical applications based targeted therapies with nanoparticles, you go back and look at all the papers. you will not find many of these which have actually gone to clinical trials, right? Not yet. So that is one of the greatest challenges with nanomaterials that I'll be talking ahead. So where nanomaterials have actually proved have gone to a clinical proven stage, those are two of those cases I'm going to discuss with you in the case studies, which have actually gone to the clinical stage. Okay. And these are the different biomedical applications regarding imaging, preclinical studies. So targeted therapies with nanoparticles have mostly been studied in case of preclinical study. But if you go to clinical studies, there are studies with these certain kind of moieties where you will find not exactly nanoparticles, but you will find radioactive labeled nanoparticles. So FDG, which can be used for PET. So when you go to PET therapy, you will find polymeric nanoparticles, micellar structures, which are being used for therapies. So some of these particles have actually been FDA approved now. Okay. But then that is only for PET scan. Regarding MRI or ultrasound, it is still developing and they are still in clinical stage. A lot of these nanoparticles are still in the experimental phase.
Okay, ma'am. Okay, thank you.
Going ahead. Yeah, thank you. Please feel free to ask questions anytime. I'm happy to answer. So going ahead from targeted therapies with nanoparticles, this is, as I said, this is a very developing field. And when I'm talking about different modalities like MRI, ultrasound, it is still developing. But if you go and try to read about nanoparticles with PET, there are some particles which have gone into FDA approval now. Now, and when I say FDA approval, there are very, very few nanoparticles which have gone into FDA approval. Most of them are still in clinical trial. Smart drug delivery systems. Now, when I say smart drug delivery systems, that means you are developing nanomaterials. Again, this is a very blooming field, which has always been been developed, and you will see a lot of papers on this, that you are developing nanomaterials which are responsive to the environmental stimuli like pH or temperature, thus allowing the controlled and targeted release of drugs. One of these examples, you'll see that there are a lot of graphene oxide, iron oxide, gadolinium oxide nanoparticles which are again tagged with either PEG, which change their shape with pH, or which are pH sensitive, which would release these drugs at more acidic pH, as cancer cells have more acidic pH than normal cells. So it is more pH responsive, and even temperature in cancer cells is higher than normal cells. So these drug delivery systems, one of the most common drugs that you'll see in papers is doxorubicin. And again, it is a drug that I worked with when I was working with my graphene oxide systems. I tagged graphene oxide using hyaluronic acid with doxorubicin, and then I studied that how the release is happening at different pH. Now, this allows controlled and targeted release of drugs. That is why we call it smart, because it is not going to release the drugs as soon as it is injected. It is going to find out whether it is a cancer cell or it is a normal cell based on the pH or temperature. So this approach enhances therapeutic efficiency while minimizing adverse effects. Again, targeted therapies with nanomaterials as well as smart drug delivery systems, you will find a lot of papers out there, many research articles. So these are some examples that you see, like carbon nanotubes, iron oxide nanoparticles, gold nanoparticles, mesoporous silica nanoparticles, liposomes, dendrimers, micelles, and quantum dots, which are again functionalized. They can be functionalized in different ways, and then they are loaded with drug, as you see. Okay, so they are first functionalized and then the drug is loaded on top of it. One of these examples, as I was saying, was graphene oxide, right? So that's what we did, like graphene oxide, and then we functionalized it using hyaluronic acid, for an example, and then we added doxorubicin, which is a chemotherapeutic drug, and then the drug delivery is studied in the cancer cell based on the stimulus. But this is for PDT, PTT, and of course PTT. So apart from drug delivery and therapeutics, now this is something that has been clinically, some of these examples are clinically approved, and I'm going to show them in the case studies. So when we go to monitoring and wearables, nanosensors have been used to enable continuous health monitoring by detecting physiological changes at nanoscale. One of these examples is in diabetes, where glucose is increased in case of diabetic patients. So they can measure biomarkers, enabling early intervention in managing chronic diseases and enhancing patient management strategies. Integrating nanosensors into wearable devices allows for real-time health tracking, providing patients and healthcare providers with valuable insights into health status and trends, which can facilitate proactive healthcare management. Very, very huge words. But then, apart from this very simple example that I'll be showing in the case study later on, is diabetes, where you can do glucose monitoring and thus send that signal to your smartwatch, and then you can say, okay, your glucose has increased, and sometimes it is automatic, like you get the insulin injection automatically, or so the nurse or you take the injection by yourself based on the timing that or the drop in the glucose level. So monitoring and wearables, since it is one of the easier part, right? It is not something that is being injected in you directly. So that is why the FDA approval is much more easier. I'm sorry, I'm continuously saying FDA because I'm working here. So most of the work that we do needs to be FDA approved. So there, for different companies, for the different countries, there are different organizations for approving these drugs, which can be injected, for approving the monitoring devices and the wearables. So in US, it is primarily FDA. So that is why I keep on saying FDA approved. So here are just some examples that how the flexibility and comfort has increased with the different generation of the monitoring and wearables. And as you see, portable devices have come into being, and now there are attachable devices to your body. You must have seen an example where these monitoring devices are, non-invasively, like minimally invasively, they are attached to the arm so that they can be monitored. Ingestible devices which you can ingest, and endoscopy can be done using these kind of ingestible devices. So because of nanomaterials, the nanomaterial technology which has been developed through electronics, through this bioelectronics that you are seeing, through drug delivery. So all these applications, because of nanomaterials, it is revolutionizing the field of medicine, as I have shown through these examples. Any questions till now? Because after this, I'll be going to the case studies. Any questions?
Other than, ma'am, other than graphene, whether there is a high value studies in other elements, ma'am, in nanoparticles?
Gold nanoparticles, silver nanoparticles. So as I said, I'll just go back to the previous slide. See, so there is gold nanoparticle, iron oxide nanoparticle, carbon nanotubes, and graphene. Of course, graphene oxide, they have gained a lot of traction in the last few years, right? Whenever you hear graphene, there's a lot of research going on graphene. And obviously, because it is non-metallic, it is highly biocompatible, so it has found a lot of traction. But apart from that, a lot of research, when I'm talking about photodynamic therapy, gold nanoparticles have been used a lot for photodynamic therapy. In fact, silver nanoparticles are being used. Iron oxide nanoparticles are being used for MRI and MPI. I hope I answered your question.
Okay, ma'am.
I'm happy to take more questions. I have reached mid of my presentation, so I thought I should let it open for the entire field.
Ma'am, can you explain that electronic structure of graphene and why it is more useful in as a nanoparticle in various fields?
Okay. So I have not added graphene oxide here in this presentation, particularly. Okay. But then, interestingly, I have another slide which is open right here. And I'm happy to take you through that. Do you see my slides?
Yes, ma'am.
Okay. So, is it okay if I go to graphene oxide, because graphene oxide is just the oxidized form of graphene, and I can talk both about graphene and graphene oxide?
Okay.
Okay, ma'am.
Yeah. So, graphene has this two-dimensional structure, and it is this integral structure framework where it has all carbon which is attached to each other. And what happens is, in case of graphene, there are different wait, I'll show you this. Yeah, I'll just show this slide. I'll just go ahead and show you the slides. Okay, so when you talk about graphene oxide and graphene, now in case of graphene oxide, there are these different oxygen containing functional moieties. I'm sure a lot of you may have worked or have read about graphene oxide as well. And this is same for both graphene oxide and graphene, that it has very high aspect ratio and it is very hydrophilic, and there is ease of processability. So you do not need to have very big instrumentation for processing graphene and graphene oxide. And so this is something that I did for my study, where I ball milled graphite, and I was able to introduce these defects in ball milled graphite. In fact, what happens with ball milling is that it goes into a graphene kind of structure, as you see this. And then the graphene structures, they are exfoliated, and then they are oxidized into graphene oxide. One question. When you are, yes.
You've told me that you have ball milled the graphene. So what was the previous size before ball milling, and what was the size that you have obtained after ball milling, ma'am?
Okay. So first of all, I'll have to go to the next slide to show you those numbers. Okay.
Yeah, sure.
Is that okay with you? Yeah.
Yeah, ma'am.
So, yeah. So, I should have kept those slides here. I didn't know people would be so excited about graphene. Anyways, it looks like this is not in these slides. But yeah, I'll just give you a ballpark number, okay? So in this case, the graphite was bulk graphite that we had bought, okay? And then after ball milling, here you see for different ball milling, for let's say if I've ball milled it for 10 hours, 20 hours, 30 hours, you see that, right? Yeah. So after ball milling it for different hours, for ball milling BMG 30, which was ball milled graphite 30, for this I had got a defect density, you can say, when I say defect means how big the size was. Okay, and we had seen it under TEM, and the size of one continuous structure of graphite, it was around 30 nanometer.
Okay, ma'am.
Do you get me? Okay. And then for sorry, for 100 it was 30 nanometer, because it is a much more complicated complicated phenomenon which occurred here. Okay, in short, I would say that when you start ball milling, let's say for 30 hours, it goes on breaking, and these defects create these very, very small graphene structures. Okay. And then after 30 hours, what happens is these small structures, there is something called annealing. Even in metals, when you see, when you go on breaking metal, it'll go on breaking, but after a certain time, when you are giving the same force, it starts annealing. So it does not break anymore. In fact, because of that energy that you are providing, they start annealing. So they start attaching to each other. So that is what we saw in my study as well, that after 30 hours of ball milling, because of the ball milling energy that we are providing, because of the mechanical energy that we are providing, a similar kind of phenomenon is occurring in ball milled graphite, that is, they are starting to anneal. So as a result of it, the 30 hours of ball milling, they were of the size of almost 5-6 nanometer. A continuous, if you look at entire sheet, a continuous sheet would be of 5 to 6 nanometer. But then when you go to 100 when you go to 100 hours, it was almost 30 nanometer. I have that somewhere in my PhD thesis defense and my PhD colloquium presentation. So this was just
One
another
more question, ma'am.
presentation.
One more question, ma'am. For every
Yes.
10 hours, you have checked the size of the graphene?
So no, we didn't. We did TEM only for 30 hours and 100 hours.
Okay. For TM for 30 hours was 30 nanometer, right?
No, for 30, that's what I was saying. For 30, when I say continuous, you do not say size. When you look under a TM, the continuous pattern, okay, and it changes.
Fine.
So you will find a defect right there.
Okay.
Between two structures. As I said, like it's somewhere in my PhD thesis defense presentation where you have those TM structures. In fact, you'll find that even in my paper, the paper that you are seeing here, Mohanta Z et al. Scientific reports. So you'll find that figure even in that. So we have those, structural defects shown in the TEM images. I know this is rather interesting, and of course, like it is more interesting for me as it was my thesis work.
And one more question, ma'am. May I know what is the type of balls used for ball milling?
Oh, these were steel balls.
Steel balls.
Stainless steel balls. Yes.
Okay.
Ma'am, can we have the DOI and the chart box?
Okay, this one? Okay, sure.
Yeah. Can we use CNTs for the same purpose, ma'am?
CNTs?
Yeah, carbon nanotubes.
Yes, in fact, yeah, if you go back and see some of my paper, I mean, not my papers, but some of my lab mates' papers, in fact, one of them did in this also. Just too many things open. Okay. I hope this is helpful. Or in fact, what I can do is, if you do not get the paper, it is open access, but just in case you do not find it, please drop me a message on LinkedIn. I can send you the PDF. Okay.
Okay, ma'am. Thank you. Thank you so much.
So I'm just sharing this as well so that you can find the papers that I have worked on. And if you look for any future collaboration, because all of you are faculties, so that's also welcome. There. You'll find the paper there as well.
Actually, I just thought of asking you the Google Scholar ID that you have shared. Thank you so much, ma'am. Thank you so much.
Okay. Yeah. Thank you. And I'll just close some of these windows so that it's not that confusing. Okay, so graphene oxide and graphene, I know it has a lot of interest from a lot of people because there is a lot of ease of processability there. And in fact, if you go on ball milling it to very, very small sizes, you'll even be able to get quantum dots. So not through ball milling, definitely there are different chemical reactions for getting quantum dots, but then you can get different structures from this. As one of the examples you said, that you can use carbon nanotubes as the precursor, and from there you can ball mill it and you can get different structures from it, using which you can put it to use to different kind of applications. So using these compounds that I showed you, I was able to find out the MR images and the relaxivity at 3 Tesla MRI scanner. And the images that you are seeing right now are the T1 weighted and T2 weighted MR images. And in this work, I was one of the work that I shared, not that one, but there was another paper which came after that, in which I was able to show that graphene oxide R1 and R2 relaxivity, which are the MRI properties, are dependent on the oxidation level of graphene oxide. Thankfully, those slides were open and I was able to tell you something about graphene and graphene oxide. And in fact, my lab mates, they did a lot of work in IAC on graphene as well as carbon nanotubes. So if you look at those papers also, those they can be very helpful. In fact, I told you, like, feel free to drop a message on LinkedIn if you want some kind of papers related to particular your field of interest. I would be happy to help out with that. Okay, going ahead. I hope, are there any more questions? Then I can just go to the case studies now. Okay, I believe there are none. You'll have more questions later on after I finish with the case studies and impact. And after that, we'll have an open discussion about nanomaterials and what do you think about nanomaterials, and what do you think about nanomaterials, and what do you think is the future of nanomaterials? I would like to hear from you as well, because let us do a little bit of brainstorming. It's not just about me going on telling about, oh, this is so great about nanomaterials and this is what is happening, but rather than how much you already know and how much we can build upon that knowledge. And let's say if you have an idea, then how feasible it is and how can you take it forward. So let us do that, because it is a faculty development program. It's not just a regular presentation that I'm giving here. So the first case that I want to discuss with you is for nanosensors for glucose monitoring that I was talking about. So glucose monitoring has been vital for diabetes management. What has been the traditional method are finger pricking, and so there has always been discomfort with finger pricking, and there is always a time lag in finding out what the glucose level is. And all those small glucose monitors that we use, they are necessarily not I just want to ask a question. So the PPT earlier that I shared, were you able to see that, the graphene oxide nanoparticles?
No.
Okay,
No, madam.
I'm really sorry about that. I didn't realize that I was only sharing my PPT, current PPT. Else okay, fine. Okay. Give me 1 minute. And I was looking at my PPT and talking to you people. I am going to share it here so that you can see. Okay, now can you see this?
Yes, ma'am.
Yes, ma'am.
Okay, this is what I was showing in the previous PPT, but I think it wasn't recorded. So this is the graphene oxide structure that I was talking about, and it is a very planar structure. And this is the graphite and ball milling graphite. I'm really thankful to you people. I was just discussing about it, and you really understood what I was trying to get to. And this is what I was telling, that the methodology that I used, that graphite to ball milled graphite, which was then exfoliated into graphene sheets and then later on oxidized. I hope you get an idea that what I was talking about. Okay, so I'm not going to jump now. I'm going to really stick to nanosensors for glucose monitoring. And so glucose monitoring, diabetes has become so common. If you look, if any person in your neighborhood or in your family, you go and ask, there are so many people who are suffering around us from diabetes. So it is a very common problem if you look at it. So now what nanosensors are offering are that you get real-time and non-invasive solutions. Now, when you say non-invasive, in case of sweat, yes, non-invasive, but in case when you are trying to take blood samples, it cannot be non-invasive. It is, yes.
It is not in presentation mode. If you will
Still?
Presentation mode.
Oh, okay. Is this okay now? Fine?
Yeah, fine.
Okay, thank you. So nanosensors, they are being able to offer now real-time and non-invasive solutions. How do they work? So glucose detection can be done through enzymatic or electrochemical methods. And they can use nanomaterials such as carbon nanotubes and gold nanoparticles. Now, each word that I'm using here is a paper on its own. So when you search these keywords in Google Scholar, that, okay, glucose detection using enzymatic reaction or enzymatic method, or glucose detection using electrochemical method. So these keywords, whenever you are trying to search on Google Scholar, because that is our greatest tool as researchers, right? As faculty, as researchers, when you are trying to write a review article, when you are trying to write a proposal for your study, what is the major setback that we have? Finding the right keywords to find out that what we need to find out. So when I say, okay, I want to find out about nanosensors, but then nanosensors is such a huge field, right? So now let's say glucose detection using enzymatic method, or glucose detection using electrochemical method. Then it could be carbon nanotubes, it can be gold nanoparticles. So you be specific when you are searching. So if you give specific keywords, try to avoid any connecting words, because then it confuses the search engine. Do not use connecting words, just give proper words, and then like carbon nanotubes or gold nanoparticles, so that it is being able to search exactly what you want. So how do these nanosensors work? They can either sense glucose in sweat or saliva or interstitial fluid. So based on what it is sensing, the type of the method through which it is sensing it differs. Now, what are the advantages of using nanosensors for glucose monitoring? They provide high sensitivity, real-time monitoring, non-invasive wearable designs, and patient convenience. There are many examples now in real world. For an example, technologies by Abbott, which has come forward, which is Freestyle Libre. And then there are emerging devices like contact lens sensors and wearable patches. What are the challenges that you face when you are trying to design nanosensors for glucose monitoring? You have to check for biocompatibility, you have to look for long-term stability, you have to find out the calibration accuracy, cost, and then scalability. Since I worked on optical glucometers myself during my MTech project, developing a simple optical glucometer itself has its own challenges, where it was an invasive technique where I had taken blood and I was trying to find out glucose in the blood. In case of sweat, saliva, or interstitial fluid, what you are doing is that now you develop this method using a material. Let's say, for an example, I'm taking gold nanoparticles. Okay. Now, at the experimental level, how do you do it? You are seeing, let's say, for an example, saliva. Okay, we are taking saliva. Now saliva has you cannot saliva from different, you can do it at preclinical level, you can do it at uh, proof of concept level, proof of concept where you're not trying to go to you are not trying to go into mice. When I say preclinical, it is mice and rat studies. Okay, when I'm saying clinical, it means human studies. So when I'm not trying to go to mice or rat and I'm trying to go to or human studies, I'm just trying to prove it at a very proof of concept level. Okay, now one way is that you take saliva from different humans and you try to find out, but that would be the final stage, right? So how would you do it? Think about it. If you are trying to develop a nanosensor for glucose monitoring, what would be the first step? I would really like your answers here. Let us make it more interactive. What would be the first step that you would do? Shoot some answers, 1 or 2 answers at least. Okay, so the first step that we would say we would do is decide what nanomaterial you want to start with, right? So my favorite material here is going to be graphene, let's say. Okay, now graphene is my material. Okay, now I want it to act as a sensor. Now think yourself and tell me that if I want to use graphene as my sensor, it should be able to sense something and it should be able to give a certain result, isn't it? It should be able to give a signal which can be detected. Now, what would be my next step? So my next step would be to take yes. Okay.
Modifications on graphitic carbonatite so that it can do the sensing.
Yes.
Like
good.
Preparing some core shell, incorporating with some metal nanoparticle. That thing we can consider.
Yes, of course. So basically, you start with your, there are three main components, right? One is what your input is going to be. The input could be a sweat, saliva, or interstitial fluid, right? So our input is the glucose in these different fluids. And then in middle, we have the middle block. The middle block is your nanosensor. As you said, now you plan, excuse me. Now you decide your nanosensor, that what nanomaterial it's going to be. And as you said, I said that let us start with graphene. And you said that, okay, let me do some surface modification. Let me create such structure which has not been done earlier. So for this, what as a researcher or as a faculty, what your next step would be? To do a literature review, right? That what has already been done.
Yes, ma'am.
Because it's of no use if you are already developing something which is already there. It is wastage of resources, right? Because it has already been developed. So basically try to do a thorough research review when you come up with an idea that this is my material. Take that material and do a literature review that, okay, this work has been done or not. And once you find out that, okay, yes.
Selecting the sensor element, before that we have to get into the review, no? That literature review.
The thought, it is the thought process, right? You have to think of something, isn't it? If you are just going through the literature review and then trying to think about this, first the chicken came or the egg? It's a simple question, right? There are two methods through which you can go. Either you go through that entire literature review and you try to find out whether, okay, what is missing? You find out what is missing and then you try to create something. Another thing is like here my friend who said that, you know, they will modify the graphene structure. Okay, now they have come down to graphene structure already. Now they will do literature review about all the graphene structures that are available. And then they will say, okay, this has not been done with graphene structure. And so this is what I would do. Understood?
Okay,
because
ma'am.
as I said, this field is so vast and the knowledge is so vast that if you go to find out what particular problem you want to solve, it is going to be a needle in a haystack. So you really need to bring it down to a certain level in, bite-sized pieces where you can actually be able to solve those problems or develop a system. And then, okay, fine, that's great. Okay, through literature survey or whatever way, you came up with this very novel kind of structure that can be actually developed. What would be your next step? First, developing it. Either I can do these reactions and you can, through chemical reactions, and that becomes your novelty, and that could be your paper, right? So that is one thing, that either develop that nanomaterial in-house, or, you design it, or you get it, you buy it, or you give some company to do it for you. And then when you ask company to do it, your novelty lies in developing the further part, getting all the preclinical work or the proof of concept work done. So the next part would be what you are trying to sense. Okay, for an example, glucose, we are talking about a graphene based nanosensor. Now our output, what our output is, my next question is, what do you think our output is in this case?
It will give some what percentage of glucose is
It's a
present.
very simple answer. Yeah, it's a very, no, it won't give you glucose. You won't get the signal as glucose. The input is glucose, definitely. But you won't get the signal as glucose. It's a very simple answer. And when I say, you will be like, oh, yes.
It's a percentage. The percentage.
Ma'am, we have to calibrate that value as per the
calibration comes
results.
later on. You went to directly fourth step. You get an electrical signal, right?
Okay.
Yeah, you will get an electrical signal. It will be form of some current or voltage or some resistivity value, right? It is going to be electrical parameter, isn't it? It's a very simple answer. So you get the result as an electrical parameter, right?
Right.
Let us say current. Let us say it's a current. Okay, just to keep it very simple. So my input was glucose. My nanosensor detected the glucose and it converted it to a electrical signal, which is in my case current. Okay, now let's say if I'm getting that current, now for different glucose levels, I'll get different currents.
Current values.
Different current values. Now when I'm getting different current values, then you go to the calibration stage, right?
Okay, ma'am.
So then after that you calibrate, and then you get this whatever relationship you find. It can be a linear, it can be it can be a polynomial. So there are different types of fitting. So you can get different equations for different kind of systems. It's not necessary that your system is always going to be linear. I'm sorry, the weather has got to me. It has snowed today in Baltimore. So yeah. So I was saying, like, after calibration, what do you do? What would you do after calibration?
We will have a reference level, no?
Yeah,
level.
then you take unknown samples.
Yes,
right?
ma'am. We will
Then
test that sample, different samples.
Yes. Yeah, now you take unknown samples and then unknown levels of glucose, and then you find out that how much of error you are getting. Isn't it? And it should be error level which should be which should be detectable, as well as it should be very, very low. We want the error to be very, very low. We can't allow very large errors. Otherwise, it cannot be used further. It cannot be commercialized. Right? So these are different steps in which a nanosensor is actually developed. Most of the information is right there. It's more about bringing all that thought process together in order to develop something. Okay, so now based on what type of nanomaterial you are taking, what kind of study you are doing, so there are so many different parameters. Even glucose, you can try to find out glucose in different ways. It can be either in sweat, it can be in saliva, it can be blood, it can be interstitial fluid. So your input can differ, your type of nanosensor can be different, your type of output can be different. Depending on what type of output you are getting, you want to detect, that also differs. So there are different components of a nanosensor, and based on what you want to detect, how you want to detect, and not just detection, the novelty also lies in how you are developing the nanomaterial, right? If there is a nanomaterial which has already been developed, but you have developed a method through which you have improved those physical and chemical properties of the nanomaterial, that also holds novelty, right? And when you finish all this work, at the end of the day, you have some papers and a patent. So that I'm talking from the point of view as a researcher. I want an end goal, right? I just don't want information out there, because whatever information we are giving here is also available on internet. You can find it everywhere, right? But whatever information we have here, if it is not turned into something tangible, which can be actually used. Hello?
Ma'am?
Yeah. Sorry, I thought I got disconnected. Everybody can hear me, right?
You can hear, ma'am.
Okay.
Your voice is perfect.
My voice, everybody can hear my voice?
Yes, ma'am.
Okay, thank you.
Yes, ma'am.
So the point, my point is that all this information that I'm sharing with you can be found on internet, right? If you go to find presentations on biomedical applications, if you go to try to find review articles on nanomaterials for biomedical application, there are so many types of resources available out there. So through this workshop, let us try to get something tangible out of it, right? If research is an integral part of, first of all, I hope that all of you are getting some understanding of what nanomaterials are, what are the different types of nanomaterials out there, how these nanosensors are being developed, how these nanosensors are being developed, and how they are being put to use, how they are being tested. Okay, so if you get that basic understanding of all the processes, and at the end of the day you go out with something tangible in your mind, that yes, this is what I can do, and this is what I can go ahead and I'll be able to do in my lab or in my institute. So and so let us try to get something tangible out of this workshop. So, as I told you, that the real world examples are like Freestyle Library, where they have developed these minimally invasive glucose monitoring devices. So, using nanosensors for glucose monitoring is already being done. And this is one of those examples where nanosensors have been used to develop a clinically approved biomedical application. So, going ahead for the case 2, with okay, I missed the future part. So what is the future for nanosensors and glucose monitoring? It can be integrated with Internet of Things and artificial intelligence for predictive analytics, development of non-invasive sensors, and improved diabetes management outcomes. So these are very important terms, because this is where the development is going on. You just don't want it to go to the smartwatch. You want that all that information can be collected, as a result of which all that information can be integrated together, and you can get all this public information through which all this data, public health data, can be developed. So that becomes really useful for the future. Not just that, if a person who is under diabetes level, like they are still not diabetic but are pre-diabetic, in those cases also, like if AI is being able to predict, and through IoT we are able to predict that, okay, if these fluctuations are being seen in glucose, or this is the trend which is being seen in glucose growth of the glucose levels, then these are the preventions that we need to take right now. if the food could be monitored, that what we are eating, what are the glucose spikes. So all these things are going to help in the future of developing these glucose monitoring devices. But of course, when you are trying to develop certain things, we really need to take some very, very important factors, which I was talking about: biocompatibility. You need to think about how stable it is along the long term, as well as we talked about calibration right now, which was a very important step in developing a nanosensor. So we need to think about what is the accuracy of calibration as well. Cost and scalability. Coming in India, especially, we know that cost is a very big factor. So again, while developing a particular instrument or a monitoring device, we need to take care that the cost should not be higher than what is already available in the market. Then it is of no use if you are getting something at a very cheap cost in India, and you are trying to build something for which the cost is extremely high. Then it does not really make sense. And of course, we can do all these research as proof of concepts, and you can have papers, but at the end of the day, if you want something very tangible which can be used in the market, but I'll tell you something, it's not necessary that the research that you are doing, everybody says that it should be commercialized, you do something which should be commercialized. The basic research also is extremely important, because basic research, when you're trying to do these basic research, it can always branch out into different fields. If you are developing a different electrochemical method, if you are trying to improvise on a particular electrochemical method, it can be taken forward, and it's not necessary that it is going to be used in biomedical application. My point is that there are various aspects of research, there are various aspects of writing papers, there are various aspects of making a patent. It all depends on what is your interest and what you would like to develop on. So going ahead, I would like to go to the case 2 where gadolinium nanoparticles have been used in MRI. So gadolinium, there are 4 or 5 companies which have gadolinium nanoparticles for MRI contrast agents. So for magnetic resonance imaging, there are 2 types of contrast agents: T1 contrast agent and T2 contrast agent. Gadolinium nanoparticles are mainly used as T1 contrast agent. Is everybody awake? Hello?
Yes,
Ma'am, we are
ma'am.
still.
Yes.
Yeah, it was so silent. I was like confused that, okay, if people are listening or not, or I'm just going on talking. Okay, so gadolinium based nanoparticles, they have been used for MRI contrast agents for a very, very long time. And these are one of the nanoparticles which actually got a lot of traction as in biomedical application. And they are the nanoparticles which have been used for a very, very long time. So gadolinium nanoparticles shorten T1 relaxation time of water proton and thus create bright contrast in T1 weighted images. This itself, like based on T1, T2 contrast agent, I can give another talk, because as I said, MRI is a very dear field of mine, and I have worked on developing MRI contrast agents. So I can talk more about this definitely. And that in case of gadolinium nanoparticles, they can be used to create this T1 weighted contrast. And since they work as T1 contrast agent, let's say you are not being able to see part of brain cancer, which is dark and is almost similar to the tissue contrast in a normal image. When you inject gadolinium nanoparticle, due to the difference in the water content and water density of the cancer and the normal tissue, there is a difference between the relaxation between both these tissues. Because of the difference of the relaxation, now gadolinium nanoparticles will brighten that part, as a result of which you will see that the cancer has lit up and it will look white, like bright, and the other tissue, it will be like, of course it contrast will change, but it will not be as much as the cancerous tissue. So because of this, gadolinium nanoparticles have been used rigorously as T1 weighted imaging, and it is regularly used in MRI contrast. Another contrast agent is iodine based ipimidol, which is one of the material that I have used in one of my recent papers. And again, iodine-based nanoparticles, we do not call it nanoparticles, we call it small molecules because they do not form nanoparticles. In the audience, can somebody tell me what is the difference between a small molecule and what is a nanoparticle?
A nanoparticle is below 20 nanometer.
Okay.
Small means maybe micro.
Those are microparticles.
Less than nano.
No, good. See, no answer is a wrong answer. Okay, there is no problem. Feel free to give any answers. Yeah, so
volume ratio.
Yes, so that is correct. It has very large. Next, any answers? What is the difference? My question is, what is the difference? What is a nanoparticle? When do you call a particle as nanoparticle? When do you call a particle a small molecule?
So when surface to volume ratio is very high, it's nanoparticle, and when surface to volume ratio is not very high, we can call it small molecule.
When that is not there, then it's called a bulk particle, as far as I understand.
Below 20
when the size of
particle size is in nanometers range.
See, for nanoparticles, you are giving all the right answers. Believe me, all the answers that you are saying is correct. That is what a nanoparticle is. Any one of the dimension less than 100 nanometer, 20 nanometer, it is nanoparticle. Okay, when you say that one of the dimensions is in nanometer, right? So it is a nanoparticle. But I'm asking about small. You all are correct about nanoparticles. Small molecule. So nanoparticles are aggregations of molecules. It is not single molecule, right? How do these nanoparticles form? There is always a seed, and then that seed grows, right? So let me say, let me go back to that iron oxide one. Okay. So if you see this figure here, if you see
there
this
must be
figure
some
here,
capping agent for nanomaterial.
Yeah, so for nanomaterial, it is not a single molecule, right? For single molecule, there are many molecules which come together to form a nanoparticle, like ice. If I take ice for an example, right? Now, how does ice form? It has a seed and then it grows in all directions, right? Similarly, for nanoparticles also, there is always going to be a seed and then it grows. It is not a single molecule. It is always a collection of molecules. Imagine it. Just like this iron oxide nanoparticles. It is not a single particle, you see. There are lots of iron oxide particles which have come together and which have formed this iron oxide nanoparticle.
Molecules can make a nanoparticle.
Yes, many molecules. What is a molecule? Molecule is just a structure of atoms, right? If you go to basic chemistry level, what is molecule? We say molecule is just molecule is a structure which is formed because of different atoms, right? Like for iron oxide, Fe2O3 is a molecule. Fe3O4 is a molecule, right?
Hello, madam.
Yes.
Sorry for interrupting. Can I know how long, how much time you need to complete this session? Because the other resource person is waiting. I'm sorry.
Oh, how long was I supposed to give this presentation?
7:30. Till 7:30, madam.
7:30, right? And what's the time now?
Yeah.
I'm on time, right?
It's
like 2 minutes?
Yeah, it's 7:30, right? Yeah, 2 minutes. Yeah.
Fine. Okay, I understand. Like, anyways, this was a very basic question. When I say small molecules mean individual molecules. Okay, so the effect can be created by one molecule itself. And these molecules are independent of each other. So that is why they are small molecules. And nanoparticles means many molecules, they come together to form these aggregates called nanoparticles. That is the basic difference. Okay. I'm extremely sorry because of my cough. Okay, so gadolinium, they have a very potential toxicity. They are very nephrotoxic. And that is why we are developing these different nanoparticles for MRI contrast agents. Future direction is to get biocompatible coatings of gadolinium oxide. Not just that, we are developing different kind of small molecule based and nanoparticle based contrast agents for MRI. So there are various challenges that we face for nanomaterials. They can be nanomaterial safety, biocompatibility issue, the scalability and regulatory issues, and long-term stability issues. And there are many future directions for nanomaterials. It can be precision medicine, advanced diagnostics, regenerative medicine and tissue engineering, immunotherapy and vaccine delivery, artificial intelligence, theranostics, nanorobotics in medicine, fighting antimicrobial resistance, brain and neurological application, biocompatibility and safety optimization, real-time health monitoring and applications in aging and longevity. As you see, nanomaterials and biomedical applications has many future directions. So it all depends on our brainstorming, that what we want to do, where we want to get to, what is our input, what is going to be the system that we want to use, and what is the output that we want. These are the various acknowledgements that I wanted to give, and thank you so much.
Ma'am, I just want one question, ma'am.
Yes.
This one iron oxide MRI agent, where iron oxide it's a magnetic and oxide material. Gadolinium also means, what are the main differences from gadolinium has from the iron oxide for MRI?
So gadolinium is paramagnetic in nature. And so it is the different type of magnetism that they have. Gadolinium is much larger than iron oxide, and gadolinium oxide, again, it is a more of a molecular structure. If you go back and look at a gadolinium oxide nanoparticle and an iron oxide nanoparticle, and gadolinium oxide, it is paramagnetic in nature. And that is why it acts as a T1 contrast agent. And if you go to iron oxide is super paramagnetic in nature. Since it is super paramagnetic in nature, it acts as T2 contrast agent, and it is very small in size. Okay.
Zina ma'am,
thank you, everybody.
Can I have one question?
Okay. Thank you, madam. Thank you for the interesting and inspiring and knowledgeable session.
It depends on the organizers.
It's time, actually, because many of them have asked the questions in the middle itself. Actually, they were supposed to ask in the end, because the other resource person is waiting, madam. I'm sorry.
Sure. I didn't take more than my time, right?
No. It's exact.
Okay, thank you so much. I hope everybody was benefited by it, and you can connect to me on LinkedIn, and I'm happy to answer any questions there. And thank you to the organizers for inviting me. Thank you so much.
Thank you, ma'am. Thank you.
Please share your mail ID, madam.
Okay. We'll be starting with the second session today. I'll just introduce the speaker of the second session. Dr. Nitin S. Kale obtained his MTech from VNIT Nagpur in 1999, PhD from the Electrical Engineering in the field of nanomaterials at IIT Bombay in 2007. During 2001 to 2007, he worked on nano cantilevers with material coatings to carry out the research project. He is currently working as R&D head, Nanosniff Technologies, Bombay. His interested areas are nanomaterials, MEMS, material characterization of biosensors. He has over 50 publications in international journals and conference proceedings and has granted 10 patents. I welcome you, sir. And today's topic is nanotechnology, microsensors, and instrumentation.
Okay. Hi. Am I audible?
Yes, sir. Good
Yes.
evening, sir. Yes, sir.
Yes,
It's
you are.
Yes, sir. Audible, sir.
Yes, sir. Audible.
Okay. So, first of all, thank you very much for inviting me. Second thing is, I will just try to upload share my presentation, that is, and then we can get into the talk. It is possible that yeah, a little bit of what I am going to speak is perhaps it has been already spoken by a previous user, but we will try to sort of build upon it. What I would like to know is the full screen visible?
Presentation is
no, sir. Not
Yes.
it is. No,
your
sir.
face
Yes, sir.
is
Yes.
slide mode.
My
Please
face.
keep in presentation mode, sir.
The screen is
visible,
not
sir.
in full screen mode.
It is not in full screen mode, is it?
Yes. The screen is visible, but not in full.
Okay, understood. Let me see. Try to figure out how that is done.
Actually, I think you are sharing the window for presentation.
Okay.
You should select whole screen.
Okay.
In sharing.
Yeah, I'll do. It has been a long time since entire screen is what you're suggesting, right? Okay. I hope this works. Are you able to see it?
Yes, sir. Just go to your presentation.
Okay. How about it now?
Yes, sir. Okay. It is on full screen. Okay.
Oh, good. Okay. So, as it would have been mentioned, that my talk is basically upon nanotechnology, microsensors, instrumentation, etc. So it is in two parts. I am going to speak more about the science and technology part today, fabrication of sensors, for example, and I will deal with the actual applications. For example, you will see nano sniffer explosives detector. So this is a product which NanoSniff, our company, has developed, and it has been installed at several airports now. So I'll be talking a lot about this and some other work that we are doing, but I'll talk about it tomorrow. So as you can see on the screen, we are a company that was incubated at IIT Bombay. We work from the research park office there. And I have, for example, Kapil, who is looking after the business of the company. Ram Gopal Rao and Soumya Mukherjee are partners in the company. They were from IIT Bombay. Now Ram Gopal Rao is VC of BITS, in group of institutions, and Soumya Mukherjee is director at BITS Hyderabad. Anup Prabhu is also a director here. So I have given a little bit of outline about NanoSniff technologies. What I'm going to do in this talk is going to talk more about nanoscience and nanotechnology from our own perspective. Then about MEMS sensors, actuators, couple of examples. I will delve deeper into nanofabrication technology today, and maybe I might touch upon micro cantilevers. So tomorrow I will deal with cantilevers and micro heaters and nano sniffer and a lot of other MEMS applications. So somehow this is working a little bit slowly. I'll need to figure that out as well. So, as I said, NanoSniff is a fabless microsensors, instrumentation and IoT company at IIT Bombay. We are into MEMS device fabrication, high speed and high specification electronic instrumentation. We do a considerable amount of bio work as well. We used to do. Now it has been reduced a little bit. We do a lot of analytics, which includes signal processing, statistics, and neural networks. In terms of MEMS technology, we are good at making micro cantilevers of the piezoresistive variety, micro hot plates or micro heaters, and we are also making micro heaters with interdigitated electrodes and metal oxides. So the key product is explosive trace detector. We are also working on gas detectors. So in terms of nano phenomena and nanotechnology, I don't know why this is not working properly. My slide changing is giving an issue. Anyways, so in terms, as the previous speaker was, very nicely discussing about the dimensions. So I will just take from whatever she has spoken, and as we are all aware, we have dimensions which are in meters. We call them macro, like our cars, we ourselves, human beings. Then we have the mesoscale, which is in, let's say, millimeters, even maybe up to a fraction of centimeters, like snails, the rice grains, ants, etc. Then we have the microscale, and this is something which we discovered somewhere in the 19th century. It includes cells, bacteria, pollen grain, dust particles, etc. And then we have the nano-size structures, which includes viruses, the nuclei, proteins, and other biological stuff. Any question? Is there any question? Okay, so I'll continue. Materials behave very funnily at the nanoscale. This is what is depicted in this slide. So first thing is, if you have two objects which are having, let us say, masses which are m1, m2, which are spaced about, let's say, by a distance of r, then as per Newton's laws of gravitation, you know that the force between them is proportional to product of their masses, inversely proportional to the square of the distances. And in case these masses are charged, then the electrostatic attraction or repulsion is governed by the Coulomb's law, which again says that it's proportional to the product of the charges on the two masses and inversely proportional to the square of the distances. Now, for a moment, imagine that you have reduced the masses by half. So what is going to happen is, it's going to reduce the gravitational force between the two, distance being the same. But these masses can still hold the same amount of charge, right? And therefore, the electrostatic attraction or repulsion is not going to change. And if you go on doing this, you go on reducing the mass, the gravitational force will become more and more redundant, and the electrostatic force is still going to remain the same, assuming that it's still holding the same charge, which is quite possible. So what we are then saying is, at the nanoscale, we do not worry much about the gravitational forces between two particles. Second thing, as far as the physics is concerned, we need to take recourse to Schrodinger's equations to describe the mechanics or the actions between them or the interactions between them. And instead of using the typical classical laws, so we cannot use them, we use quantum mechanics. A good example is, many of you are from the electronic side, I know that, and the gate oxide thickness of a transistor, which used to be several tens of nanometers a few decades back, is now about 1 nanometer. So at that dimension, the tunneling happens, right? So the charges are tunneling through now the dielectrics. And therefore, why this is happening? This is happening because of the physics or the quantum mechanics at the nanoscale. And therefore, the solution for that engineering wise is we are using high K dielectrics like hafnium oxides. The previous speaker spoke about greater surface to volume ratios. We can have a quick slide about that very soon. Another important point is random molecular motion becomes more important. For example, in very confined spaces, each interaction between the, let's say, the walls of the space can create a different force as compared to what is happening in a large box, and there are gas atoms moving here and there. What you can also see is what are called as the colors of nano gold. So what you will see here is that the material is the same, which is gold, but the material is existent as a colloidal solution, and the material is in different bottles. It is of different particle size. What it tells you is, what you can see is that the color of the colloidal solution is different, and this is only because the size of the particles are different. And therefore, what we are then saying is, by tuning the particle size, we can tune the optical property. And this is very, very important sort of exploitation of the particle size in terms of the optics. Okay. Again, I believe you would have seen this slide, but let me just repeat this. This is the impact of surface to volume ratio. A lot of you are definitely aware of it. So what you see here is that you have a cube which is, let's say, 1 meters by 1 meter by 1 meter. It is having a certain volume, 1 cubic meters. It is having a certain surface area which is 6 times 1 meter squared. Right? Now what you do is you split it into half along the 3 axes. What happens is that the volume remains the same, but the inner surfaces are now exposed, and therefore the exposed surface area increases. You go on doing this, the volume still remains the same, and the surface area is going to exponentially increase. What this tells us is now the bulk material, what is inside in terms of, let's say, the mass, that is no longer important when you have the material at the nanoscale. Rather, it is what is present on the surface. What is the chemistry on the surface? What are the bonds on the surface? These are going to play a much more important role as compared to what is inside the bulk. Now, nanotechnology is not really new, actually. So a few examples, 1, 2, 3, 4 examples I'm giving. For example, people used gold nanoparticles to make glass, the ancient Romans. People used gold and silver salts to make the glass of church windows in the medieval times. There are examples of hair dyes, which Greeks and Romans used to use using a certain material. And even the Indian traditional bhasma preparations, they are shown to contain nanoparticles. Please remind yourselves of bhasma or the Ayurveda, wherein you have the traditional mortar and pestle, and the person is actually crushing some material to a certain particle size. Now keep in mind that if a particle size is going to determine its functionality, and if you have the wrong particle size, then what is medicine could become poison, and even vice versa. Okay. Again, there is a issue with my, okay, this is worked. Okay. Now we'll go to some of the laws which are called as scaling laws. Again, if you are electronic engineers, you would have seen that a few things happen in VLSI design, which is why we have actually scaled the gate length of the transistor over the last couple of decades. Advantages in terms of density of devices, in terms of area required, small, in terms of power required, in terms of speed of operations, etc., etc. All that is depicted here. What I like you to focus on is, let's say, an example which is, let's say, heat flow. So across small objects, heat is going to flow very fast. That is again something which I'll show. Frequency is inversely proportional to the length. This again is of very of a substantial importance in MEMS devices, and of course the other stuff you can check. Okay, in terms of volumes and in terms of some other constants that you see on the left hand side, you know that 10 centimeters by 10 centimeters by 10 centimeters is 1 liter. That is 1 liter is, let's say, the amount of milk that we buy every day. That's a 1 liter pouch, let's say. Now, if you divide it by 10, all dimensions, you have 1 centimeter by 1 centimeter by 1 centimeter, and that becomes a milliliter. So 1 liter is 1000 ml, and 1 cc now happens to be 1 milliliter. If you further reduce that by again 10, then you have from a milliliter you go to a microliter, where you have the dimension as 1 mm by 1 mm by 1 mm. Now, why all this? The idea here is that tomorrow, if we are going to build an example I'm giving, let's say a lab on chip, then whatever medical tests that we require to do today with pathology laboratories, typically we give 5 milliliters of blood. The same test could be done using a lab on a chip by, let's say, 100 microliters or maybe just 1 microliter. That's possible. So that is one of the very important driving force in nanoscience and nanotechnology. Other example which I can give you, how serious this becomes is for young people, like many of you students, giving 5 ml of blood once a day, no problem, maybe even 10 times a day, no problem. But think of people who are on the older side, people who are unwell. For them to give 5 ml or even 10 ml, because multiple tests need to be done every day, is very, very difficult. And therefore, you can see that this is a very important driving force. Okay. Some of the small scale phenomena that people have observed, for example, you can see the example of a bulb here. This is an electrical bulb with a tungsten filament. This typically takes about 75 to 100 watts. The filament goes up to 2500 degrees Celsius, and it takes about 20 milliseconds for it to reach that temperature. I'll give you an example of micro hot plates tomorrow, where the same thing happens. You go from, let's say, room temperature to 500 degrees Celsius at few milliwatts. So, where you have 75 watts and where you have a few milliwatts, right, in terms of requirement. And the temperature change is huge. And this happens within, let's say, 2 to 3 milliseconds. One more example is from the biosystems, where, it's very, very hard to actually create balls between two chemicals in biological systems. So typically these are going to intermingle. And the reason for that is because mass is going to flow very easily through thin barriers, unless some sort of energy is required to prevent the movement. Please keep that in mind whenever you are doing biology. Okay. Some of the examples in terms of small scale phenomena you can see on the left hand side. You have a micrograph of a stainless steel material, and you can see grains, you can see grain boundaries. But when you look at stainless steel with the naked eye, you see it to be very strong, right? You are confident about its strength. You are confident that it can bear huge loads. However, when you look at the micro scale, you realize that there are grains and grain boundaries. And then you understand that when a material is going to fail, it is going to fail along one of these grain boundaries. The other thing that you can see on the right hand side is an example of hydrogen bonding. The gecko or the lizard, as you as we call them. Lizards are known to climb walls. They are known to do it because of forces of hydrogen bonding or the van der Waals forces, because they have certain very small structures on their palms which are called as cilia, right? And humans have now been able to fabricate these cilia-like stuff. You can see things on the left-hand side, for example, here. So this is something that humans have made. Okay, so we are trying to mimic what is there in biology. Okay, so these were some of the examples which dealt with nanoscience and some of their examples. Now let us switch gears, move on to the engineering systems. What you can see here is a violin. That violin is having several strings, and you can see the size scale. This was made about 30 years back at Cornell. The point is that rather than having just material, powders, you can actually now build devices, and somebody can play this with an appropriate pin or with an appropriate gadget, and it will resonate, it will create a sound. Whether we will be able to hear it or not depends upon the resonant frequency. So with this small example, what we can say is that when a system is engineered, then we use materials, not just one, but multiple materials. We try to build components, we try to build devices. Using various devices, we build subsystems, or we might call them as modules, and eventually we assemble a system. And this is what all engineered systems are all about. Example, you can see on the left hand side an automobile, and on the right hand side the human body. So let's compare. So you have components, you have materials. Let's say if you take an example of tire, then you have rubber. Rubber is today fed with carbon nanotubes. Then you have components. I have given the example here are certain shock absorbers which are having springs. Then you have sort of a module, which is either the brake module or the transmission module, or maybe the sort of assemblies, different assemblies inside. And eventually all these subsystems are then assembled into the automobile. Let's take what is happening on the right hand side. On the left hand side is something which humans have made. On the right hand side is something which nature has made and the evolution has made. So what you can see here are different systems. So a human being having the circulatory system, the digestive system, the nervous system, etc. And then if you go down, you could have an organ like, let's say, a kidney, and then you can have cells. Then you can have inside the cell you have DNA, RNA, etc. And then you have the structural building blocks, the proteins, which are again further made up of amino acids. So that is the typical one is the top down, which is the car. Second is the bottoms up, which is the human body. Okay, MEMS. The previous were the examples of how engineered systems are made. Now we move to microelectromechanical systems or microsensors, which is what NanoSniff is into. So we use the word micro electromechanical systems, which means that there is an electrical and electronic stuff inside. You have mechanical, so there is going to be some sort of a movement, or it could have some sort of a heating activity. They are used in terms of different names all over the world, like microsystems tech or micro machines, etc. And when the dimension becomes smaller, then you have the NIMS or the nano electromechanical systems. Okay, now the one slide on the commercial side. Let us say you have your mobile phone. So what do you see? You would see something like a microphone, which is a MEMS device. You will see a speaker, which is a MEMS device. You have accelerometers, you have gyroscopes, you have compass. All these things are MEMS devices. Okay, and this is just an example of what all kind of MEMS devices are present currently in the world. For example, you have your filters, you have your oscillators. So everything today, except the memory and the logic, which you called as CPUs and GPUs, etc., a huge amount of sensing has moved to the MEMS technology. And these are few examples. In terms of markets, these are today billions and trillions of dollars kind of markets. For example, you have pressure sensors, microphones, gyroscopes, in terms of their numbers and their forecast. So something like around 20 billion this year is the forecast for just MEMS. The numbers are indicated here. So yeah, there is good amount of scope as far as the career is concerned of students. You can see that there are certain market trends in which people are interested. For example, you can see consumer devices like Fitbit, which is employing again certain accelerometers on one side, and then you can see certain medical devices like hearing aids, right? They are also going to have some sort of a speaker some sort of a membrane to pick up the sound and then transmit it to the ear inside. So these are two different directions. On one side you have consumers, and one side you have medical devices, and then you have a sort of a marriage between the two in terms of different products. So you can see again that in terms of consumer healthcare, there is a considerable amount of interest in these devices. I think this we have discussed, or maybe this is a different slide in terms of how things have grown over the last 6 years. I will skip this slide. Again, this is how different sort of markets or different products are giving revenues, and you will see that certain products which were very important, sort of their growth is sort of stable and not further increasing, but then there are certain products whose growth is increasing exponentially. Particularly, for example, this brown thing here. So this is growing considerably high. So in terms of what MEMS devices are, so what they do is something, let us see. So they sense something or they actuate, they will try to compute some certain things, they will try to definitely control some things, they will communicate, and it is possible that they can also power. MEMS borrows a lot from the integrated circuit technology, that is the CMOS VLSI technology. For example, batch fabrication, miniaturization, mask process, then sort of the marriage of IC and MEMS, IC technology and MEMS. So these things are directly taken up by the MEMS technology, and it is a joke that whenever the machines become old, the IC industry gives it away to the MEMS industry. They are quite small. The scale is present here. So typically from, let us say, 100 micrometers to 1 micrometer. So 100 microns to 1 micron is the scale in which typical MEMS are present. And then in terms of, since it is very close to 1 micron, then the wavelength of light is also very close to 1 micron. So MEMS devices also function as, you would have heard the word optical MEMS or MOEMs. So those kind of devices have also been made with the MEMS technology. This is an example of what kind of a PhD level research can be done in the area of MEMS. So let us have an application that we want to build tweezers which can pick up a cell from a desired location, and we can place the cell at a desired location. application, in terms of application, you might be, the scientist or the doctor might be interested in looking at a certain tumor. So certain tumor is to be studied. So what you see here are micro cantilever structures which can be actuated so that a small particle such as a cell can be actually picked up, and once it has been picked up, it can be placed wherever you sort of want it. And then each device actually can be a part of a much larger system. You could have for example certain motors which are going to move it. You could have the electronics which is going to connect and allow you the electrical actuation. So all those things are possible. And then you could have a scientist who is working using this particular preset. One of the first products of MEMS was an accelerometer. It is present along the airbags of an automobile. So it started with the driver getting protection for an automobile, for a car. Then it moved to the passenger sitting beside the driver, and then now it's very soon going to become compulsory even for the rear passengers to have airbags. I'm not talking about the seat belt, I'm talking about the airbag. So airbags are becoming compulsory for the rear sitting passengers as well. So you can see where you had a market of 1, now you have a market of at least 5. Okay. So how do these accelerometers work? So you have a proof mass which is suspended. So it is moving along with the car with a certain velocity, and a part of the proof mass is held, right? So for example here it is fixed, this part is moving, right? So what happens is that when the car hits, then you have a certain relative motion change, and whenever there is a relative motion change, you are going to have a change in the resistance, because as you can see here, this is a piezoresistive type accelerometer. You could have a capacitive accelerometer as well. So in that case, the distance between the plates is going to change, and you can have a voltage output. In this case, you can again similarly have a voltage output. So that is going to actuate the airbag, and it's going to cause it to inflate within, let's say, a millisecond or two at max. So great application, has earned lots of money to the MEMS industry. MEMS became even more important because of the micro motor application, particularly because 2 types of joints could be made with MEMS device. One is similar to your elbow joint or the revolute joint, and second is called sort of a shear joint. It's called the sliding link or the sliding joint as well. And this was achieved by what is called as freeing the movable layer using sacrificial micromachining. I'll explain what that is in a couple of slides. But what you do is you sacrifice a layer so that you can create space for the part to move in which you are interested. Okay, so that is the basic idea. How that is to be achieved, we will see very soon. During the 70s and 80s, there was a rush to make a lot of MEMS devices. These are some of the examples. Inkjet printer head. Nowadays we generally use laser printers, but inkjet printers were very popular maybe 2 decades back, and that technology is still very valuable. So, and it is going to find different applications as well. It's a great technology. You had micro mirrors, you had microfluidic devices, you had microbolometers, then you had switching devices, you had pressure sensors, so on and so forth. So in a typical MEMS device, as I earlier said, you will have one part which moves. Okay. Then you are going to do a bulk or a surface micromachining so that this part is going to move. You give space for the part to move. And then you have certain contacts and electrical connections. You can power it. and you can read from it, so on and so forth. Here is an example of, again, MEMS, but what is called as packaged MEMS. So what you can see here is that on the right hand side, you can see that the MEMS device is very small. Please pay attention to just this area. This wire that you can see is the electrical connection. Okay, so I'm just giving an example. You could have multiple such wires. Then you have this fellow coming out, and you have some sort of contact pads here or pins or something like that, right? So you do your wire bondings here. Then this entire thing that you see here is the package. This is a jelly which is sort of protecting here, but it could be different in different devices. But what you see here is that package of a MEMS device is very, very important. It is as important as the device itself. And the reason for that is because of the packaging, you are able to interface your MEMS device with your world, that is the macro world. You're able to do mounting, you're able to do fittings. In case of bio devices, you can introduce samples through the fittings, maybe pipes. You could have optical windows for the modems. Heat management is done. You protect it from dust, etc. There could be damages due to handling. You protect your device from that as well. So a lot of solutions, real world solutions, are achieved because of packaging. And you can see, so nicely and neatly the MEMS device has been packaged inside. This is an example of a digital micro mirror. This was invented by Texas Instruments somewhere in the 70s. It is basically it's a DLP projector basically, right? So what it has are mirrors. You can see here, these are tens of thousands of mirrors. Okay, it is going to have some sort of a aluminum coating which is highly reflective to create the mirror effect. The mechanical part is the beam here, so this can undergo torsion, so it can move up or down. And finally, you have certain electrostatics, electrical connections. When you apply a voltage to that, you will be able to achieve them. So you have mirrors, you have the hinge for it to do a mechanical movement, and then you have the electrodes where you apply voltage. Please focus on the word addressable. So when I say addressable, I can address each and every mirror. And what I can do is when I sort of move the mirror, then I can determine whether the light is going to be reflected onto the screen or not. Okay. So this was a very successful product, as important as the accelerometer. Texas Instruments made a lot of money on this. The third example that I'm quoting is from the printing technology. So there are two different types of MEMS devices. I will sort of give or explain each device. Having a bit of water. Okay, so this is an example of piezoelectrics. You know what piezoelectrics are. So in piezoelectric, basically by applying a voltage, you change the dimension of the material, or if you change the dimension of the material, you will be able to measure or at least generate a voltage across the material. So that's the idea. So here what you see is there is a reservoir which is filled with ink, and there is a piezoelectric element. What you do is you apply a voltage to the piezoelectric element. Let us say it is pulled. So when it is pulled, there is a space that is created here, and ink is going to be drawn into this reservoir. And then you reverse the voltage. So now the expansion of the material becomes a contraction, and it immediately pushes that ink drop. Right. So this is the mechanism by which piezoelectric printers work. Please note what is there inside. you have a nozzle, you have a piezoelectric element which is micro machined. It is moving, therefore MEMS, right? So all those MEMS items are here already. The other method is a thermal inkjet printer. What the same thing that you did with piezoelectrics is achieved by heating as well. So what you do here is you heat it up. When you heat it up, it's going to expand, and then ink drop is going to jut. And after it has jutted, then you cool it down, and therefore new ink is going to be drawn into your reservoir. So it's as similar to what you saw earlier, but the sensor is different. So instead of a piezoelectric element, you have some sort of a micro heater. The physics of an inkjet printer is very, very interesting. Not only do you have to worry about the density of the fluid, the mechanical properties of the fluid like the surface tension, because that's going to determine what is the drop size of the drop that has been ejected. Then these drops have to break up. Okay, it doesn't have to go as a single squirt on the screen. Then you should know what is the distance between each droplet. Then you should know if air is moving, because the drops are so light, the motion of the air can affect it. Since there is a gap between the nozzle and the screen, there is a possibility of gravitational force also to be pulling it in a different direction. And therefore I said that the physics of an inkjet printer is very interesting. This is again what I discussed. I'll skip this slide. Now, if you want to make an inkjet printer device, what will you do? You need to address the inkjet printer device, therefore you need a MOSFET. So you see, you have silicon, you have source, you have drain, you have the gate dielectric, you have the field oxides, etc., etc. Right? Along with that, you have the contacts and you have the isolations. Then you have the ink barrier, right? And then you have a sort of a nozzle here. So along this, you can actually eject the ink. So this is one device, but then you can have millions of them. Okay. So what we have covered so far is we have looked at nanotechnology. We have looked at MEMS, the commercial aspect. We have looked at the basic MEMS devices. We have looked at some of the more popular MEMS devices which have made money. Now what we will do is we will look at the MEMS materials and processes. This takes you into even certain nanofabrication technologies. Example, you have old materials and old processes. Silicon is an example. SiO2, silicon nitride, that optical lithography, it's an old process. Silicon is an old material. CVD method, chemical vapor deposition, is an old method. Doping is again an old method. In the second phase, you had the old materials, but you had certain new processes which helped in batch processing, which helped in improving the quality of the material, etc. For example, you had stuff like wire bonding and wafer bonding, etching, which was normal etching, wet etching earlier became dry etching and deep reactive ion etching, etc. You had stuff like polysilicon coming in. You had stuff like sacrificial layer processes, etc. Along with this, there was a phase 3 where you had new materials but old processes. So the same CVD, the same etching and doping techniques used now with ceramics. More metals. We are getting in polymers, let's say PDMS or SU8. Materials like silicon carbide, piezoelectric films, This is phase 3. And now actually for the last 8 to 10 years, we are into phase 4, where new and exotic materials are coming in, nanomaterials are coming in, and quite new processes like, for example, Liga is also a relatively old process, but for example, you have wafer dicing using stealth dicing. So that's an absolutely new process. You have certain EUV technologies for lithography. That's an absolutely new process, etc. Okay. So we start the materials and processes section with clean rooms. I'm not sure if you have visited clean rooms, but this is how a typical clean room is going to look like. You can see, the floor color. It's pretty always like this. You can see the walls. You can see the lighting. You can see one fellow standing here in a certain, I would say, uniform. You can see another person here, third person here, and then there are certain equipment. In a clean room, what we try to control is the particle count, because if you have particles, these particles are small size, they can fall on your substrate, your silicon wafers, and they can damage the device. You are interested in controlling the temperature because you are working with a lot of materials which are temperature sensitive, and you are working at very small scale. So because of small changes in temperature can produce small scale dimensional changes, and your device can go haywire. Also, you need to control the humidity in a certain range. Too humid, your polymers will not adhere to the substrate. Too dry, they are going to show cracks. So you need to maintain that as well. On the right hand side, what you see are silicon wafers. Silicon wafers are made from silicon boules, single crystal silicon boules, and these boules are actually made generally from the Czochralski process, where you start with a seed crystal and you have a silicon melt, and then you draw the crystal out from the melt, and because of quantum mechanics and a lot of processes of the crystallography, the material that is going to come into the wafer or into the boule is going to have orientation exactly similar to the seed crystal. Now, what do you do with a silicon wafer? An example is, you make MOSFETs, transistors. A lot of processing. You do lithography, you do thin film deposition, you do etching. So thin film deposition, for example, is an additive process. Lithography is I would say a sort of a patterning process. Etching is a subtractive process. So you do add, you pattern whatever you have added, and then you do subtractions. Whatever you don't need, you are just going to remove. This is how a device is built. This is how a VLSI device is built. This is also how a MEMS device is built. So as I said, what you can see here is are the three processes, etching, deposition, and either diffusion or growth. Of course, these are a little bit complicated processes. I'll explain them one by one. Okay, silicon to silicon dioxide oxidation. It happens in furnaces at reasonably high temperatures. It is called as a growth process because silicon is consumed. The substrate, which is silicon wafer, is also consumed. It is done in the presence of oxygen, that is the oxidizing species. Sometimes it is done in the process of hydrogen as well, because if you want a wet oxide, you use hydrogen as well. So water vapor is the source for oxidizing it. What silicon dioxide does? It forms an extremely brilliant insulator. Okay, and besides this, there is nothing which needs to be said. A superb insulator. Diffusion, you want to dope your silicon, right? You want to change the conductivity, you want to create source junction, you want to create drain junction, etc. How do you do doping? So you will have some boron or phosphorus or antimony, something like that. That is the source of your dopant. You put that dopant in the furnace. You have your silicon wafers and you open the wafer at, that is your insulator that I talked about, or silicon mask or a silicon nitride mask, dioxide or a nitride mask. So it is through this mask that the dopants are going to fuse inside or diffuse inside the vapor. That is diffusion, and the electrical conductivity is going to change because of that. Then you have thin film deposition. The difference between growth and deposition is that in growth the substrate is consumed, in deposition the substrate is not consumed. The species in gaseous phase, they are going to come, they are going to sit on the substrate and they are going to react and they are going to form a film on the substrate. For example, you can have a silicon nitride film. You have silicon as the substrate, you have silane as the incoming gas, which is going to give you silicon in deposit form or deposition molecule form, and you have ammonia, which is going to give you nitrogen. So silicon and nitrogen, they bond to form silicon nitrogen at that temperature. You have what are called as, hello? Okay, I guess it was a false. So you have some more thin film deposition methods like the APCVD done at atmospheric pressure. Again, the source gases come in vapor form or in gaseous form and they interact inside the furnace. You could have a furnace which is brought to a low pressure, so it's called as LPCVD. There would be certain pumps which is going to remove the atmosphere from the furnace. You have also what is called as plasma enhanced CVD. So what is this? So typically CVD processes, chemical vapor deposition processes, happen at high temperatures. Okay, the temperature could be something like 600 degrees Celsius. But if your substrate is not silicon, instead it is, let's say, a plastic, a polymer, then you will need to do these depositions at lower temperatures because plastic polymers typically cannot withstand high temperatures. So what do you do? You are going to get the gases, right, into a chamber, and then you are going to apply an RF power. You are going to apply electric field. Using the electric field, you will dissociate the source gases. Say silane becomes Si and H, ammonia becomes N and H, and then the Si and N can react, and that film is going to deposit on your substrate. So that is plasma enhanced CVD. You can have a thin film deposition which is in the form of physical vapors like evaporation. So you could have either a thermal evaporator or you could have an E-beam evaporator. So what do you do? You have a vacuum chamber, you have your source material which could be gold. You have your wafers which are stuck inside inside your chamber, and then you heat your board which is having the gold wire to a very, very high temperature. And because of the phase diagram for the material, the phase changes from solid to gaseous or to vapor phase directly. And the gold vapor is going to go and sit on the silicon vapor. And it's going to deposit there as a gold film. Whatever you did thermally, you can also do it with electron beams. You can do a local evaporation and you can still deposit the material. Sputtering is another very interesting method of doing physical vapor deposition, where basically you have again a plasma and you have your source material, which is one of the electrodes, and the incoming gas is sort of accelerated using the electric field, and it is made to strike your one of the electrodes, which is holding. Let's say you want to do gold deposition, so it is for gold is your sort of target. So it hits the target, it ejects out the gold material, and this material is then going to go and sit on your substrate. So you have various methods of sputtering. Magnetron sputtering is the most popular. Okay. So these were deposition techniques. Now we move on to lithography techniques. So as I said, when you do lithography, you are essentially doing a pattern transfer. And the steps that you take to do lithography is you will use a photoresist. The photoresist has to adhere after spin coating. It has to adhere on the wafer. I'll show a couple of. So I'll show a couple of slides in which it will be depicted how it is done. Using lithography and using certain mask, you are going to activate or photo activate your resist at certain areas. And after activation, you can remove the photoresist from that area. And therefore, now your etchant has an access to the underlying material which you want to remove. And after the material has been removed, you can strip away the photoresist. And therefore, you coat, you protect, you expose, you etch, and you repeat. So this is how you eventually build a 3D structure. This is the mask. This is the UV light, which is the photo or the used for photo activation. This is a UV aligner machine inside a yellow room. The silicon substrate, the material of interest which you want to pattern, the photoresist on which you have designed, let's say, your pattern. So after you send your light through this, you have a structure which is like this, which has been, let's say, weakened. And after you do the development, the photoresist is removed from this place. Now, if you use an appropriate etchant, you will be able to remove the oxide. There are positive photoresists, there are negative photoresists. So wherever the light hits the positive photoresist, it is going to weaken. Wherever the light hits the negative photoresist, it is going to become strong. And using the appropriate developer, you can develop it. So this is the pattern you will get in a positive photoresist. This is the pattern you will get in a negative photoresist. You have also something which is called as subtractive and additive pattern transfer. I have exactly explained that. There are a lot of lithography tools. One is a proximity printer. This is present at many places in India. Second, you have contact printers. So a proximity printer and contact printer typically come as a single unit. Basically, it allows you to control the distance between the mask and the substrate. In the contact, the mask is going to stick with the substrate. In proximity, it could be slightly away from the substrate. This is a very expensive tool. These are projection printers. And if you read about ASML, that's a company which makes one of the best projection printers today, that is ASML. You can definitely have a look at it. This is how photoresist is spin coated. So you have this red liquid, you drop it on the wafer, and you have a spinner, you spin it. So the photoresist is going to be uniformly coated across the wafer. Right. After spin coating is complete, you can do a baking if you want. You can sort of strengthen the photoresist. And you can see here that why temperature control of the clean room is important, because the viscosity of the photoresist changes with temperature, right? And therefore, the thickness of the photoresist is going to be different at different temperatures. So you have to be very careful. when your device sizes are so small, a minor change in the temperature will give a change in the viscosity, and all your dimensions could be lost. Now etching. So certain important methods I will explain. One is the wet etch. You use certain chemicals. It is having it, it suffers from undercut problems. Then you have dry etch, basically uses gas plasma. You can have great dimensional control with that. So today much of the semiconductor industry uses dry etch and very rarely uses wet etch, except in certain cases. The dry etch could also be due to reactive ions, for example, chlorine and fluorine based chemistries, or even bromine based chemistries in certain cases. There are certain newer etching techniques which are used to etch only silicon substrate. They are, for example, the deep reactive ion etching. You could get either isotropic surfaces or anisotropic surfaces. Anisotropic means directional control you have as far as the etching goes. And you can have very interesting patterns of the material that you so desire. I also mentioned that I'll talk about surface micromachining and bulk micromachining. So in a surface micromachine film or a surface micromachine device, you basically have a sacrificial layer. Here is an example. You start with a silicon wafer, you deposit an oxide, and then you deposit, let us say, another material, which is, say, polysilicon. You pattern it. Now, you want that this polysilicon should be a movable material, okay, a movable structure. So you put it in an etchant of silicon dioxide. Now this silicon dioxide gets removed and this polysilicon is now movable, okay, it can move. So it can, for example, vibrate, right? It's a cantilever. That's the MEMS part. In bulk micromachining, what happens is, rather than a sacrificial layer, it is, you sort of dig a hole in the ground, let's say. So in your substrate material itself, you are digging a hole. So this is your substrate material. This is the structure of your interest. After masking, etc., you have kept it like this, and you have dug in the substrate. So you can have a freestanding structure which is capable of movement, and you have dug a hole beneath it. So these are two different approaches of micromachining, so that you can free a mechanical structure. This is some sort of a cartoon which explains how it is done. Generally, hydrofluoric acid is used to etch away the sacrificial oxide. Okay, Lika is again a German term where essentially you start. So it's a method to do X-ray lithography, use or make very thick structures. So what you do is, you have a metal base, you put some sort of an acrylic material like PMMA on it. Using a mask, you create your pattern of interest, you develop your material, then you electroplate. So this electroplating gives you a very thick material of your interest. You remove the PMMA and you have the thick material. So this could be, for example, a gear. One more technique which is used in the IC industry is called as planarization. What happens is that because of micromachining, many of the structures that we create become what people call as rough. It is not rough to the naked eye, but these are micrometer level eruptions. So for that, they use planarization methods. Basically, there will be a very a chemically active slurry, and there would be some sort of a rotational motion carried out, using which the wafer or the substrate or the surface is going to be flattened or planarized. Wafer bonding is the method to stick two wafers. It could be done either through thermocompression, or it could be done through some sort of anodization or electroanodization. It is very necessary that the surfaces are very, very clean. They're flat. Only then the adhesion is good. And the wafers have also similar TCs. So one method is anodic bonding or field assisted thermal bonding. You heat it basically. You apply a voltage across it, and the sodium flows inside, and you have a bonding between the silicon and silicon dioxide, and the two wafers are then going to bond with each other, the glass and the silicon. The thermocompression, you need high temperature, and also you need pressure. So it is just based on these two that the two surfaces are stuck. It has been known that the anodic bonding is much stronger because there is a chemical bonding involved rather than the thermocompression, in which there is just a glue which plays the. Okay, so these are my last few slides now. So I've talked a lot about materials, I've talked a lot about technologies, I've talked about growth, deposition, etching, etc. Let us see how we make an electrostatic motor using surface micromachining. So it's going to look like this when it is built, and when you remove the sacrificial layers, it is going to move. It is going to be like this. So poly 1, poly 2, poly 0, these are all electrodes, and this is the rotor part. This is that rotor part which I was talking about. Okay. So how it progresses is, you have a starting material, you deposit poly 0, you pattern poly 0, you deposit oxide 1, you create what are called as dimples in oxide 1. These dimples are certain features which prevent stiction from happening. Okay. Then you etch your oxide, you deposit poly 1 layer, you etch poly 1 layer into shape which is like this, you deposit oxide 2, then again now your cross section is looking, let's say, something like this. You have your oxide 2, second oxide is like this, this one is now looking like this. The rest of the etchings we have already carried out. Okay, after you etch oxide 2, you deposit poly 2, you etch poly 2, and finally you deposit and etch the metals. Now you again, remember, this is the first slide that I showed you. It's something like this, and you need to now remove all the sacrificial oxide. So you see it in cross section, and once you remove the sacrificial oxides, you have all your electrodes, you have a rotor, and you have a stator, and your micromotor is ready. So this was a very popular project in the early 90s, making micromotors. Imagine tomorrow if all the motors or the moving parts inside your watch, inside your wristwatch, are changed. You can imagine what's the market size. Okay, so how much time do we have, sir?
Hello.
Yeah, how much time do we have?
Hello, sir. It's actually from 7:30 to 9. So last 10 minutes is for question answer session.
So I have another 7 minutes. So I'll quickly deal a little bit with our work on micro cantilevers and micro hot plates. Okay.
Okay, sir. Okay.
So this is how a micro cantilever looks like. It is stuck at one hinge. It is free on the other end. There is a length, there is a width, and there is a thickness. The thickness is very small as compared to the length and width. So you have one active area on which you can do a lot of interesting things. For example, you can stick something. The resonant frequency will change because it's a function of the mass. You can carry out a chemical reaction, right? And if you carry out a chemical reaction, it's an exothermic change. You could have a bending in up and down direction because of the reaction, or you could do antibody antigen interaction by modifying the surface of the cantilever. You can have a surface stress. In a cantilever, you could have either compressive stresses or tensile stresses. Accordingly, the cantilever is going to bend. The bend cantilever is going to have a certain radius of curvature and a certain deflection. So these are standard equations which have been there in mechanical engineering and physics for the last 100 years plus. Bimetallic effects are also known. Based on the difference in temperature coefficients, you can have a change in deflection. This is again very famous formula for the springs, the spring constant and the resonant frequency, which, as you can see, depends on the spring constant and the mass. So if there is a mass adsorbed, the spring constant is going to change and the resonant frequency is going to change. In terms of detection, a bent cantilever can be either detected optically. So you could have some laser focusing on it and you collect on a photosensitive detector, or your micro cantilever could be a part of a Wheatstone bridge, and one cantilever bends, there is a change in resistance if it's a piezoresistive technique that we are following, and then you are measuring the change in resistance with your electronics. So you could have one as a reference cantilever, you could have one as the measurement cantilever. Right? So all these things are possible in terms of detection. These are some examples. I'm not exactly sure I should get into this, but this is an instrument that I built for, or a tool that I built for depositing thin films of nitrides and polysilicon and doping it also. This is called a hot filament CVD setup. These are some of the cantilever structures that I had fabricated with the films that were deposited in the hot wire CVD setup. We did some FTIR kind of analysis on it as well to verify that we had silicon nitride films. We also carried out crystallinity measurements. What is its orientation? What is the crystalline fraction? What is the grain size? What is its electrical conductivity, etc., etc. So I guess, Madam, Sir, I would like to stop here and maybe give time for question answers, because we will get into an altogether different topic, which I will prefer to handle tomorrow.
Any questions from the participants' side?
Hello.
Hello.
Hello. Yes.
Hanji, that's sir. This was a very informative session. Sir, I have a question that how can microsensors integrated with IoT enhance real-time healthcare monitoring and patient outcomes?
Just please, could you please repeat your question?
Yes, sir, sure. Sir, how can microsensors integrated with IoT
hold on.
enhance real-time healthcare?
Yeah, hold on. We'll come to that. Microsensors integrated with IoT. Okay. So typically IoT system is going to have three parts. One is the communication part, second is the computing part, and third is the measurement part. Okay. The measurement part is the part which is your sensor, which is going to interact with the real world. Okay. Now this sensor has to be connected electrically, I would say at this point of time, electrically connected with your computing, and your computing is then very easily connected with the communication. Okay, now as far as the real-time part is concerned, so as long as you are monitoring your sensor in real time with the help of your computing computer and communication, then your job is done. Have I answered your
Great,
question?
sir. Thank you, sir. Thank you.
Did
Yes,
I
sir.
answer
Yes,
your
sir. Thank you.
question?
Thank you.
Okay.
Yes, sir. Yes, sir. Thank you.
Okay.
Hello. Any more questions from the participants? Okay, sir. I think no more questions.
Okay.
Thank you, sir. Thank you, sir, for your interesting, inspiring, and knowledgeable sessions on nanosniff technologies, MEMS sensors, micro cantilevers, micro heaters, and the fabrication methods, sir. Thank you so much.
Okay. Thank you so much. Thank you, everybody. We'll see you tomorrow. Bye.
Okay, sir. And tomorrow's session will start from 6 o'clock. Thank you all.
Okay.
Thank you. Okay.
Ma'am, is there any feedback session and all, ma'am? Feedback form and all?
Feedback form, it will be at the end of the workshop on Saturday.
Okay. Okay, thank you.
Ma'am, please share the PPTs.
Yeah, definitely. As soon as the resource person shares his PPT with us, we'll share it with everyone. Definitely, all the PPTs will be shared.
At the end of this session or this end of this FDP, you can share the recorded sessions also.
Yes, we can. Okay, this recorded sessions, everything will be uploaded in the YouTube.
YouTube, okay, ma'am. Okay. The document session, the Atal portal document session also, I think you can upload PPTs.
Yes, there also it has to be uploaded.
Okay, thank you.